In the last lecture, we learned that energy stays constant in a system, but is transferred from one state to another to power pistons, shafts, fans, or other elements to turn heat into work. Thermodynamics is what powers our world and our cells. Instead of using heat to move parts, we turn food into the fuel that drives every cell in our bodies. And although we generally think of engines and power plants when we think about energy, every cell in our bodies uses the laws of thermodynamics to power itself and accomplish the tasks for which it has evolved. Whether it's to pass along information, as is the case with brain cells, or to circulate oxygen, as blood cells do, or any number of the other thousands of jobs that our cells accomplish every day to keep us alive. Whereas in the last lecture, we talked about thermodynamics from the perspective of mechanical engineering, in this lecture, we turn to the chemistry of thermodynamics to investigate how heat, or another source of energy, can turn into work. How a cell transforms matter and energy to do work is called metabolism, from the Greek for change. In this lecture, we're going to take a closer look at the power generators in our cells and those of plants. Our goal is to understand how cells turn sunlight or food into a form of energy that can be used to do the necessary work. We'll be delving deeply into the chemical factories that power our bodies and all living organisms. The chemical transformations known as metabolism follow very specific steps or pathways, and cells have evolved ways of speeding them up or slowing them down, depending on the need. What if we could harvest the sun to power our world? It's a nearly infinitely renewable resource that will only disappear when our star burns out, taking our world with it. Well, the truth is that we're already there. Photosynthesis, or the conversion of sunlight into sugar and other chemical energy stores, provides sustenance for almost every living thing on Earth, either directly or indirectly. Even the fuels that we burn to heat our homes and power our cars have their origins in plant and animal life that put the sun's energy into storage hundreds of millions of years ago. Of course, it would be wonderful if we could get all of our energy from the sun directly, although personally I'd hate to give up chocolate and other delicious foods. But our cells have evolved without the capacity for photosynthesis. What makes plant cells so special? The answer lies in part in their structure. Our ability to move around the world seriously impedes the ability to capture energy from the sun the way that plants do. Our cells are squishy and mobile. Plant cells are much more stable and structured. And this architecture is what allows them to take advantage of certain properties of a specific molecule, chlorophyll. So how does a plant turn sunlight into energy? In essence, plant cells take carbon dioxide molecules from the air and water molecules from the soil or other places and add the energy of sunlight to make glucose or sugar. Then they either use the molecules of extra oxygen and water in other reactions or expel them for our consumption. You'll notice later in this lecture that this reaction is essentially what our cells do, but in reverse. We use glucose or sugar, add oxygen and water to it, and create the energy that our cells need to work, plus the byproducts carbon dioxide and water. But how exactly does sunlight become sugar? Like its name, photosynthesis largely consists of two components. First, the light reactions which capture energy, the photo part, and require sunlight. And second, that energy is then used to build glucose and other molecules, the synthesis part, called the Calvin cycle after the person who discovered it. These are also called the dark reactions because they don't require light. Both of these processes happen in an organelle called a chloroplast. That's unique to plant cells. Chloroplasts are similar to the mitochondria that power animal cells that we met in the second lecture on the building blocks of life. All eukaryotes, remember, those are cells with their nuclei enclosed by membranes, have mitochondria, but plant cells also have chloroplasts. As I mentioned earlier, the structure of chloroplasts is the key to their success, so it's worth understanding how they're built. Surrounding the organelle is an envelope of two membranes like the nucleus in a eukaryotic cell, and inside it is a fluid called the stroma. Within the stroma is a third membrane system made up of sacs called thylakoids. Thylakoids are often stacked in columns called grana. This structure, while highly efficient, simply wouldn't be possible if plants were mobile the way animals are. It's flexible enough to bend a bit, but it's just too rigid for walking, running, and jumping. Chlorophyll, which is pigmented green and is the star of the photosynthesis show, is found in the thylakoid membranes. Now, as we discussed, the light reactions convert solar energy to chemical energy, and this happens in the thylakoids where the chlorophyll is. The Calvin cycle happens elsewhere in the stroma. But how does light contain energy in the first place? Quanta, or packets of light, travel like waves, and we call them photons. Photons have no mass, but a fixed quantity of energy depending on their wavelengths. Light with shorter wavelengths has more energy in its photons than longer wavelength light. Things that absorb light, as opposed to reflect or transmit it, are called pigments. A pigment's color depends on the wavelength of light that it absorbs. When white light shines on a plant, places that are high in chlorophyll look green to us because chlorophyll absorbs violet blue or short wavelength and red longer wavelength light, but reflects and transmits medium wavelength green light. When a photon of light hits a molecule of chlorophyll, one of the electrons in the molecule is elevated to an orbital of higher potential energy. As we've learned, electrons travel around the nucleus of an atom with specific routes. These routes are called orbitals, and each shell or layer of electrons in an atom has a specific pattern of orbital routes. These orbitals force negatively charged electrons to travel far away from the nucleus to avoid all the other electrons that repel them. So the orbitals that involve travel further from the nucleus make those electrons easier to pick off. We talked about picking off electrons when we discussed electricity. Now, the jump from one orbital to the next takes a specific amount of energy for every different molecule. That's why only certain wavelengths are absorbed by certain molecules, and why chlorophyll can absorb photons from violet and red light, but reflects green light. The closer the orbital is to the nucleus of an atom, the more stable it is, because the electron is attracted to the positive core of the nucleus. So when an electron is excited and jumps up to a higher orbital, it can't stay there very long because now it's in an unstable state. So outside of a chloroplast, the electron would just drop back down and the potential energy of the photon would be dissipated as heat or light. But if the chlorophyll is in a chloroplast, it's surrounded by other chloroplasts, and like a game of telephone, the photon gets passed around from one chloroplast to another until it reaches a place called the reaction center complex. Now think of this center as the switchboard at the telephone company. The message has been passed around until it gets to the electronic voicemail box, and there the message is held until the person to whom it is destined can pick it up. Energy from the sun is stored in a comparable way in the reaction center complex, inside the stacked columns of thylakoids. Now you see why the static architecture of the plant cell is critical for photosynthesis. In the reaction center complex, there is a molecule that can accept the excited electron called the primary electron acceptor. In fact, there are two types of reaction centers called photosystem 1 and 2. One photosystem gets struck by a photon, which pops off an electron and sets off a series of reactions in which the electron falls back to its stable state and boosts an electron in a nearby pigment molecule. And so, electrons are bumped up and fall back down until they are finally accepted by a third type of molecule. This acceptor molecule then transfers the electron into what's called the electron transport chain, and eventually the second photosystem. At the same time, the process creates a molecule called adenosine triphosphate, or ATP for short. The second system accepts the electron from the first photosystem, while at the same time, another photon excites a second electron in this system, and these electrons are used to generate another molecule called NADPH, or nicotinamide adenine dinucleotide phosphate, that, like ATP, is a repository of chemical energy. Together, ATP and NADPH are then transferred to power the reactions in the Calvin cycle. So these initial reactions take energy in the form of photons from the sun and ultimately store it in the form of these two types of molecules, ATP and NADPH. Next, we're going to move on to the Calvin cycle, or the dark reactions. Though the term dark reactions makes it seem a little sinister, they really should be called the light-independent reactions, but that's a mouthful. They are called the dark reactions because they no longer need photons, just the products of the light reactions plus a few other ingredients. And though we're getting deep into the nitty-gritty, the main idea to keep in mind is that these reactions are the synthesis part of photosynthesis, the part in which glucose and other molecules are built. Metabolic pathways, or the series of chemical reactions that turn food into fuel, can be roughly divided into two types: catabolic, or energy harvesting, and anabolic, or energy consuming. Catabolic pathways break down matter to release the potential energy in molecular compounds, while anabolic pathways require energy to create the cell's components like proteins and amino acids. The dark reactions might not need light, but they do require energy, so they are anabolic, or energy consuming. The light reactions provide the energy, of course, in the form of ATP and NADPH. Note that the dark reactions don't happen at night. They don't need light in the form of photons, but they actually only occur during daylight hours. Let's examine what it means that the light-independent reactions are a cycle. Let's think about these reactions as though they were on an assembly line in a factory. Products are passed from one station to another, where some components are exchanged or added, and then eventually the final product is released. But the machines or the stations along the way are reused to produce the next instance of the product. We don't build new machines every time we make a new iteration of a product. That would be wasteful and prohibitively expensive. Instead, each station reverts back to its original state and more raw materials of the product enter the assembly line. In an analogous way, carbon dioxide molecules enter the cycle and go through 3 stages, the second of which requires energy. During that phase, or at that station, ATP and NADPH are added to the cycle from the light reactions to power the machines. Through a series of reactions, the details of which are beyond the scope of this lecture, the carbon dioxide molecules are rearranged in the first phase into one larger compound, catalyzed by an enzyme, a molecule that speeds up the reaction called rubisco. I know, it sounds like a brand of crackers. This first phase is called the carbon fixation phase because carbon is fixed into another compound molecule. Then the compound molecule gets energy from ATP and NADPH, and through another series of reactions, a sugar is produced. This is the output of the assembly line. This sugar is then shuttled around the rest of the plant to fuel its cells as necessary. Now that we've covered the basics of photosynthesis, let's take a moment to step back and appreciate what evolution has accomplished in this elegant process. Photosynthesis is the process by which light is turned into food. First, the light reactions capture energy from the sun in the form of photons and then use it to make ATP and NADPH. Then the Calvin cycle uses these products to create sugar with the help of carbon dioxide. The sugars then supply the plant with chemical energy and carbon skeletons so that the plant cells can manufacture all the molecules they need to perform the functions of life. The mitochondria in plant cells use the products of photosynthesis as fuel, and the excess sugars are stored in various parts of the plant, like in seeds, roots, and fruits. We'll talk about how mitochondria extract energy from the sugars next. But don't forget that humans and other animals are attracted to plants. They eat their seeds and fruits and other parts and disseminate the plant's genetic material so that new plants can grow, since, of course, plants can't move around by themselves. And perhaps most importantly, one of the byproducts of photosynthesis is oxygen, which animals like us need to survive. Plants use up carbon dioxide, which is emitted by animals during their own metabolic processes. Without photosynthesis, life could not survive on Earth. And metabolism in animals helps power photosynthesis by providing carbon dioxide. An elegant, efficient system indeed. And on that note, let's turn to how mitochondria can now extract energy from sugar, as is the case in metabolism in animal cells. Most of us encounter cells for the first time through the lens of a microscope, and in that environment, they seem to be just sitting there doing nothing, maybe moving slowly in their medium, but really they seem pretty lazy. But the truth is that what we can't see is an entire factory's worth of actions and reactions, both chemical and electrical. Cells have multiple jobs. And of course, different types of cells have different subspecialties, from muscle control to information signaling to detoxification and development. All of these jobs and the factories that create the products they need to accomplish them require fuel. In animals, food needs to be ingested and converted to energy so that the protein factories can be powered. This process of converting food into fuel is called cellular respiration. As I mentioned earlier, cellular respiration is photosynthesis in reverse, taking sugars, adding oxygen to them, then breaking them down to release energy and expel carbon dioxide as a byproduct. For the next few minutes, we're going to explore the steps that are involved in turning food into fuel. Just like in the internal combustion engine in your car, your cells mix hydrocarbons with oxygen and let out an exhaust of carbon dioxide and water. But how? The answer, once again, lies in the activity of electrons. Moving electrons around releases energy in organic molecules, energy that is ultimately used to build ATP so that that energy can be stored for later use. Cellular respiration can be divided into three main phases: glycolysis, the citric acid cycle, and electron transport. Glycolysis means splitting sugar, referring to the process by which sugar, in the form of glucose, is split into two molecules of a compound called pyruvate. It's actually a 10-step process, and we won't get bogged down in the details, but the end products are pyruvate and two molecules of energy-storing ATP. Interestingly, however, the first few reactions in the process actually require energy, just like in photosynthesis. And at the peak of the energy-consuming processes is the production of sugar, the same output of the Calvin cycle that we encountered a minute ago during our discussion of photosynthesis. After the production of sugar, the energy requirements change, and the reactions begin to yield energy rather than to use it. Even though glycolysis releases a couple of molecules of ATP, the majority of energy harvesting happens in the next phase of cellular respiration, the citric acid cycle, or as it's sometimes called, the Krebs cycle, after Hans Krebs, the German-British biologist who worked out most of the pathway in the 1930s. It's worth noting that glycolysis happens in the cytosol, or the fluid-filled medium of the cell, whereas the citric acid cycle occurs exclusively in the mitochondria. These are the energy-producing organelles that we first met in the second lecture. You might remember them as the only parts of the cell that have their own DNA, passed down exclusively along the maternal inheritance line. After glycolysis, pyruvate migrates to the mitochondria and enters the organelle. There, oxygen is added to a reaction that produces a compound called acetyl coenzyme A, or CoA for short, that then enters the citric acid cycle. Like the Calvin cycle in photosynthesis, the citric acid cycle can also be thought of as an assembly line that manufactures specific products. Acetyl-CoA enters, and then there are 8 steps that eventually result in the release of ATP and a return to the original state so that another acetyl-CoA molecule can pass through. What's truly amazing is that the cells are metabolizing food all the time. 70% of our total energy expenditure happens within the organs of our body. Only about 20% of the calories we ingest are burned off with physical exercise. Of course, that percentage varies according to your level of physical activity, but it's still true that most of the food you eat every day is used up by your cells and in just their everyday functions. Think about how quickly you begin to feel faint or show other symptoms when you haven't eaten in a while. Our cells are engaging in these complex reactions every moment of every day. After the Krebs cycle, the last step in cellular respiration is what's called the electron transport chain. Here, electrons are passed along in a series of reactions, gradually releasing energy that can be used to make more ATP, just like in between the 2 photosystems during photosynthesis. The building blocks of the chain are located in an internal membrane within the mitochondria, so that the different molecules in the chain can be held closely together. In this way, the structure of mitochondria is reminiscent of the structure of chloroplasts. Ultimately, the electrons are passed down to oxygen, the final electron receptor. Because of its polar properties, oxygen draws the electrons toward it along the chain, and because we breathe in and out all the time, there's plenty of oxygen to go around amongst all of our cells. But the electron transport chain is just the first step in this last phase of metabolism in the mitochondria. In fact, the next step is likely the most important. Whereas glycolysis and the citric acid cycle produce about 2 net molecules of ATP for use elsewhere during each cycle, oxidative phosphorylation, or the combination of the electron transport chain and a process called chemiosmosis, produces 26 to 28 molecules of ATP. For a long time, this last step, the chemiosmotic process, was a mystery. It wasn't until 1961 when Peter Mitchell, a British biochemist, proposed that moving protons across the inner membrane of the mitochondria stores energy in the form of an electrochemical gradient, that the mystery was finally solved. In a nutshell, his theory suggests that energy from the electrons in the transport chain is used to pump protons, or hydrogen ions, across the membrane, and then they move back through an enzyme called ATP synthase. The flow of protons back across the membrane provides the energy needed to form ATP. The remaining electrons and protons at the end of the pumps are joined with oxygen to form water. And there you have the whole process of turning food into fuel. Thermodynamics in chemical form. Understanding the metabolic pathways or chemical reactions in cells is not just of esoteric interest for biologists. This knowledge has now been used extensively to improve our lives. Just like the laws of thermodynamics and mechanical engineering led to the development of steam engines and cars and jet planes, understanding the chemical engineering behind metabolism has let scientists create treatments for cancer, improve agricultural areas, and a myriad of other applications. Harnessing a cell's metabolism to build products is called metabolic engineering, and the ultimate goal is to develop cost-effective and ethical ways to produce valuable substances in large amounts. Current metabolic engineering techniques involve using well-known, safe microorganisms on an industrial scale to produce bulk chemicals, such as those used in fragrances and perfumes, essential oils, vitamins, agriculture, and many more. But the future entails tailor-made designer cells that might make synthetic organic chemistry production obsolete. One of the first major success stories involving metabolic engineering is the production of an anti-cancer therapeutic agent currently used in the treatment of several types of cancer. In this story, the needs of cancer patients were pitted against the environmental costs of harvesting a naturally made compound. A chemical called Taxol was discovered in the 1960s, collected from the bark of a Pacific yew tree known as Taxus brevifolia in a forest in northern Washington state. In the late 1970s, Taxol was shown to be mildly effective in the treatment of mice with leukemia. Human clinical trials began soon thereafter, and in the late 1980s, the compound was shown to have a beneficial effect on patients with melanoma, or skin cancer, and ovarian cancer. But in order to treat all the current cases in the US, the pharmaceutical company would have to destroy 360,000 trees every year. Now, the Pacific yew tree is a smallish evergreen tree that can reach up to 10 to 15 meters in height. It grows along the coast from Alaska to California, but its growth rate is extremely slow, making farming of the tree very difficult. The problem for the pharmaceutical company that wanted to produce this effective chemotherapeutic agent in bulk is that harvesting the bark needed to make it killed the trees outright. So by the 1990s, as the environmental movement was rapidly growing in the US, and especially in the Pacific Northwest, there was pressure to create the drug synthetically, saving the trees in the process. The best method discovered took advantage of metabolic engineering and in the process eliminated the environmental costs of killing trees, and it also uses fewer toxic chemicals in its production than purely synthetic methods. It also uses less energy. Today, the drug Taxol is FDA approved for the treatment of breast, ovarian, and lung cancer, and although it's now available in a generic form, annual sales at its peak in the year 2000 reached $1.6 billion. All production of Taxol by the Bristol-Myers Squibb pharmaceutical company uses plant cell fermentation technology, metabolic engineering, and there is no longer any need to take bark from trees. Another success story was revealed in 2003 when engineers at UC Berkeley found a way to make an anti-malarial miracle drug available to developing countries. Now, malaria is a parasite, and it's become resistant to many of the traditional treatments widely available. But the treatments that can kill the resistant strain were too expensive to be distributed to developing countries. Until, that is, the team from Berkeley figured out how to splice genes from 3 separate organisms into a single microorganism and create a bacterial factory for the effective drug called artemisinin. With this metabolic engineering feat, artemisinin, a potent antimalarial drug, is now readily available in the poorest countries where it is needed the most. Metabolic engineering is now widely used in the pharmaceutical industry, but it has also improved the production of some of our favorite vices: cheese, beer, and of course, wine. Understanding the laws of thermodynamics has proven to be critical in the development of machines and vehicles, in the chemical engineering that powers our cells, and now in metabolic engineering that offers solutions to challenges we face in industrial production, public health, food production, and more. But there are many things we still have to learn. Perhaps the most mysterious is the second law of thermodynamics, that heat flows in one direction from hot to cold, or, more generally, that the universe moves from order towards chaos. So often we see organization in biology and in nature and even in our own civilization. Our cities seem to get more and more organized and less chaotic, and yet the second law holds true. Another mystery is, why can't energy be created or destroyed? What, in the end, is energy? The study of thermodynamics is unique in science in that it largely developed from applications and the need to solve practical problems rather than to answer some mystery of the universe. But even so, the second law has underlined one of the biggest mysteries of our time, posing profound questions such as, why does a system, any system, move towards a state of disorder? While the answers to these questions continue to elude us, the order observable in the laws that energy follows is remarkable. That order, in many respects, governs the way cells have evolved in order to harvest energy and use it to sustain life. Another set of laws governs a different aspect of life, both at the cellular and the macroscopic level: the way fluids behave. In our next lecture, we'll delve into the world of fluid mechanics and what it has to do with making airplanes fly.